interior of the endoplasmic reticulum, and then they drift through the rough ER, but eventually arrive here at the smooth ER. There's that protein, so let's watch it snake its way through the rough ER and arrive at the smooth ER. Then we'll see what goes on. All right, when a newly made protein arrives at the smooth ER, it has to be sent outward. It has to be sent to a new location because proteins operate at specific spots in the cell. For instance, if this was a, that's a good review question. If this was a histone protein, what part of the cell is its destination? Where does it have to go to? Nucleus, right, because histone proteins live in the nucleus and they're the spools for wrapping up the DNA and the chromosomes. If this was a receptor protein, where would it go in the cell? Cell membrane, right. If it was a transport protein, where would it go? Cell membrane also, yes. And so different proteins have different destinations inside the cell where they're supposed to be. And so this thing eventually has to get shipped outward from the smooth endoplasmic reticulum and eventually has to go wherever it is that it's supposed to go inside the cell. And so what we're faced with right now is transportation of newly made things away from the smooth ER eventually to wherever they're supposed to go. And the transportation structures inside cells have a name. They're called vesicles. And so what we're going to see here in a moment is the newly made proteins and the newly made lipids are all packaged up in a little round ball that's called a vesicle. And vesicles are formed simply by budding, kind of bubbling outward of membranous organelles. So watch this. Here's the newly made protein. Part of the membrane that forms the wall of the smooth ER just kind of bubbles outward like that. The protein goes inside there and then it pinches offward like that. And that little bubble of membrane carrying the newly made protein inside is called a vesicle. And I think that is in your notes here. Small membranous spheres, little balls made out of membrane that are transporting materials inside the cell. So think of vesicles like those semi-trucks. If you go stand out by the highway, you'll see trucks going this way and trucks going that way all over the place. If you look at a cell under a microscope, a really good microscope, you would see dozens, if not hundreds of vesicles going all around the cell, from this organelle to that organelle to the cell membrane, away from the cell membrane to the nucleus, away from the nucleus. They're just constantly, organelles are constantly sending materials back and forth to each other using these vesicle transport structures. If we just discuss the vesicles that bud off of the smooth endoplasmic reticulum for a moment, what those ones are carrying are newly made proteins, because those are made in the endoplasmic reticulum, and the newly made lipids. The newly made lipids, like the newly made phospholipids, will be in the wall of the vesicle, since that's made out of phospholipid. Matter of fact, I think the next scene is kind of a close-up of the vesicle. There it is. Any newly made lipids that are being transported, like newly made phospholipids or newly made triglycerides or newly made steroids, anything hydrophobic becomes part of the wall of the vesicle, and anything that's not hydrophobic, like the newly made proteins, are carried inside the vesicle. All right, so the vesicle is just budded off from the smooth endoplasmic reticulum. Now, if you think of, think of a big truck full of freshly made produce, like just that was just plucked out of a field on a farm. They don't take that produce right to the Safeway store. They first send it to a sorting and distribution center to package it up and then decide exactly where it has to be sent to. And the cell has something like that. That's actually the next organelle. That purpley looking thing off on the right is the cell's sorting and distribution center for newly made proteins and lipids. and it's called the Golgi body, or sometimes the Golgi apparatus. So all of the vesicles that bud off of the smooth ER, which have the new proteins and new lipids, those vesicles automatically go to the Golgi, so it can sort of sort them out and figure out where it's supposed to send those proteins and where it's supposed to send those lipids. Okay, so there's the Golgi, and there's that vesicle. The vesicle just kind of fuses with the Golgi. Maybe I should run that backwards. Remember, the walls of the vesicle are phospholipids, and so those phospholipids just fuse with, just add on to the phospholipids that are part of the wall of the Golgi. The Golgi is a membranous organelle also. That is, it's got hollow water-filled spaces here, but the walls of those spaces, the walls of those tubes are made out of phospholipids. So the newly made phospholipids and triglycerides and steroids just become part of the Golgi's membrane, but the stuff that was inside the vesicle, like the newly made protein, just end up inside there like that. Now, part of the Golgi's job, well, it receives the new proteins and lipids, but it has to sort of figure out where those newly made proteins are supposed to go to inside the cell. And they don't fully understand how the Golgi figured that out yet, but they're pretty sure they know. If this is supposed to be the newly made protein, oh yeah, so that's the newly made protein. What are the monomers? Amino acids. What they see is, for the proteins that are made inside the smooth endoplasmic reticulum, they're of course made out of amino acids because that's what proteins are made out of. But they also find stuck onto the side short chains of sugars. Each of these is like a little sugar molecule. There's a sugar molecule. There's a sugar molecule. There's a sugar molecule on the side. And what they think is those are destination tags, that when the smooth endoplasmic reticulum is sorting, it is carrying those newly made proteins, it adds those little sugar tags onto the side, and somehow that encodes where its final destination is. If you go to the airport to fly somewhere, on your piece of luggage they put the little tag saying send this luggage to Heathrow Airport in London, or send this luggage to Dallas, Texas, wherever you're going, that you get the tags on your luggage. That's what they think these sugar tags are on the side of the protein are for. And so the Golgi apparatus must have some sort of enzymes that can read those saccharide tags, and from that information figure out where this particular protein is supposed to go. So as the protein drifts through the Golgi apparatus, it's reading those tags, and I think it modifies the tags, perhaps in some cases removes them entirely, or perhaps in some cases adds some sugars that weren't there before. But in some way, it reads that information and figures out where the protein is destined to go to. And when it figures that out, you get a new vesicle forming where it's going to send it to its final destination. The protein reaches one of the tips of the Golgi apparatus, and then, just like we saw with the smooth endoplasmic reticulum, part of the membrane that forms the wall of the Golgi is going to bubble outward. And the protein will slip inside there, and then that will detach, and now you have a new vesicle that's going to carry that protein to wherever it is that it's supposed to go to. Any questions up to this point? Fascinating. All right, so where is that protein going to go to? Well, as I tried to say earlier, it could be anywhere in the cell. Depends what type of protein it is. Here's another review question. If this was the enzyme RNA polymerase, where would it go to? Anybody remember where the enzyme RNA polymerase functions? Nucleus, right. That's the protein here in the nucleus that manufactured messenger RNAs. If this was a protein that linked phosphate groups to glycerols and making phospholipids, where would it go to? The smoothie R, right? Because somewhere in the smoothie R we have enzymes that make phospholipids. If this was a channel protein, which is the same thing as a transport protein, where does it go? Cell membrane, right. All right, so this vesicle is going to go to wherever its destination is supposed to be, wherever that protein is supposed to end up. And just for the sake of illustration, let's say that it is a transport protein, and so it has to go to the cell membrane. Well, that's where it's going to go then. Let's watch that happen. And remember, the cell membrane is made out of phospholipids, as is the wall of the vesicle. So again, we just have fusion. The phospholipids that are part of the vesicle wall just become part of the phospholipids that are part of the cell membrane. And then they close up around that transport protein. And that's how transport proteins or receptor proteins or any other type of protein that's supposed to be embedded in the cell membrane gets embedded in the cell membrane.
So the vesicle, does it travel to near space on the going to the cell membrane? Will it just kind of float around or something?
For the vesicles, they do have railroad tracks, and I haven't shown them yet, but a little bit later on in this lecture on the cells, I'll show you those railroad tracks. And so when the Golgi creates a vesicle, it puts it on the right railroad track to go to wherever it's supposed to go. It's amazing. Cells are like little cities. They have transportation tracks, they have sorting centers. They're astounding little things. Okay, now let's kind of go back in time for a second and give ourselves another vesicle. Here, this is yet another vesicle that's just come out of the Golgi. And let's say that the substance, the protein inside here is something that's supposed to be exported from the cell, or I guess I should use the term secreted. Cells secrete things, and that means export them. And just to give an example of that, think of the cells that form